Yes.
So thank you so much, and thank you for Active Motif for first of all, for organizing this exciting event. It's been really great to hear many colleagues present their work, and also for the opportunity for me to showcase also the work that we are doing in my lab. And so very briefly, an introduction. So we already heard the beautiful talks earlier today, and also just the recent one from Job, thinking about how the organization of the genome in the nucleus is important. And we know that gene regulation occurs from the level of single nucleotide point mutations all the way to the position of nucleosomes, the histone modifications, binding of transcription factors, but higher order organization aspects are also important, such as the contacts between enhancers and promoters, how all stretches of chromatin are folded together or separately in terms of active states, for example, in compartment A or in compartment B, all the way to how all chromosomes are organized in the nucleus. And we heard from beautiful work from Job's lab in the association to the lamina and how this organization dynamically changes. And one reason this is extremely important is, of course, to understand in particular genetic disease. And this is just one example I think many of us show in our talks. This is an example from schizophrenia, where a number of variants have been described here. This is already in 2014. And in this particular study, the vast majority of these variants actually are found in non-coding parts of the genome. And so it becomes a mystery really to know what are they doing. Are they changing the binding of a specific transcription factor, repositioning a nucleosome, changing the enhancer promoter organization, or maybe altering the specific folding of a domain, or even altering the lamina association, and in that manner affecting genes. Now, one challenge is, of course, this one, to know exactly at what scale of regulation are the variants occurring. Another one is that we know that the gene regulation, it's very different genes are active in different cells, this we all know. But importantly, also the regulatory landscape, the enhancers, they're much more varied and cell type specific than gene regulation itself. And so not only if we want to understand the variants, we need to study and understand the folding of chromatin and all the epigenetic layers, but also be sure to analyze the cell type where this particular mutation may have its role by altering the mechanism that we want to discover. So the major questions that guide our work and many colleagues in the field is really to understand which genes are regulated by which elements in the genome, in which cells is this regulation occurring, when exactly, and aiding our understanding of mechanisms of development, but in particular also mechanisms of disease. And so in my lab, we have strongly been using a technology that is rooted on elements from electron microscopy. And initially, we were using sections called Tokuyasu cryosections, where pieces of tissue or cells are essentially just fixed and cryoprotected in a sugar solution, then frozen liquid nitrogen. And from these tissues, we can obtain thin slices of the tissue that essentially contain the molecular content of the cells in that slice. And initially, and in particular in my PhD and in the lab while we were in London, in the first part of becoming a junior PI, we essentially used a lot of this technology to understand the organization of chromatin, even at the level of electron microscopy, or the organization of specific molecules. This is, for example, visualizing sites of nascent transcription. Or all the way to looking at how all chromosomes or specific genes and their transcripts are positioned in the nucleus when the gene is on or not. However, these imaging techniques, they are, of course, very important and beautiful. They allow us to see specific genes and increasingly many genes in parallel. But essentially, we still need to choose what we want to look. And as we started to discover elements of patterns, such as, for example, here in this picture, you see elements of chromosome 5 closely interacting with chromosome 7. And we could become very excited about this event, but it's without knowing exactly what's happening in the rest of the genome, in other parts of the nucleus, it's very difficult to know whether this very exciting event is actually specific and being driven by some regulatory mechanism or just a bystander of some other folding pattern that is occurring in the same nucleus. And at this point, it became really clear that we had to move from imaging, and in particular from looking at specific loci one at a time, to analyzing multiple aspects of 3D genome topology, and also across the whole genome, all in parallel. And so this has been our driving ambition since then. And so to do this, we developed a technology that we call genome architecture mapping, where we're using exactly the same slices that still contain all the same molecules, but instead of overlaying them into a glass cover slip or an electron microscope grid, we essentially move them to a laser microdissection film. And from there on, we're going to isolate each of the slices by laser microdissection and essentially extract the composition, the molecular composition of the slices. And in the first take of this method, we essentially extract all the genome content of the slices, and we ask which bits of the genome are more often appearing in the same slice. We do this first by extracting the DNA and doing whole genome amplification, sequencing, and then a lot of bioinformatic methods to decide whether a piece of DNA is or not in the slice, followed by mathematical and statistical approaches to infer topology at the different scales. And so the way this works in a simple manner is that if you think that we are interested in the pink and the blue locus here, that is very close in a particular population of cells, then if we take this randomly orientated slices, we will very often, when we find the pink, we'll find the blue because they're so close. It's very difficult actually to slice one without the other. But of course, conversely, if we are interested whether the green and the pink are together, we will rarely, very rarely find them in the same slice, and even more rarely green, pink, and blue. And so the way this method works is a bit like surveillance and ecology, and using principles of spatial statistics to infer organization. And so the data, the genome architecture mapping data, is essentially a big table in which we have the data from different cells, from single cells, which we call from, in this case, from a nuclear slice, each from a different cell. And we then ask, you analyzing the data, which parts of the genome are present or absent. And from this data, we can then extract a number of different topological parameters, of which just one is really the pairwise contact. So what is the frequency by which any two pieces of the genome appear in the same slice? And one way to understand how this method works is really to just look at us collecting the data here. And so this is a matrix of contacts along the genome, a small part of the genome. And if we collect 13 slices, you can already see a pattern, but of course you don't believe it because there's a lot of pieces of this matrix that are not filled. And so you continue to collect, and you'll see that after 33, it's looking better, but still, you're still perhaps not fully satisfied. But then you do a few extra dozens, and you will now see that the pattern is becoming crystalline. Of course, there's still a few empty places. Maybe there's some structures here, the intense co-segregation, that it's still statistically not robust. So you continue and you collect not a few tens, but actually more often, or in most of our project, collect several hundreds of these slices, between 500 to 1,000, where we then can have some statistical understanding of the probabilities at which each locus is appearing and co-segregating. And we know from the work of Mario Nicodemi that essentially the GAM matrix is a distance matrix, and we can essentially understand that these two loci are much more often in very close distance in the genome than the same locus, for example, at a different distance here, who are seldomly coming together. Now, the matrix can also be then understood as a full description in terms of models, and I hope towards the end of my talk, I'll come to discuss this in more detail. So the advantages of the method are that we can indeed work with very low cell numbers. We typically use 1,000, but I think you could appreciate that even with less, we can extract spatial parameters. It's important because we can work with very small numbers, in particular with tissues that are very small, in biopsies. And we can extract not only the typical pairwise contacts, but also radial position and chromatin compaction. And something that I won't have time to talk in this talk, but that we can also, because all the molecules are still in the slices, quantify not only the DNA that is in each slice, but also RNA molecules, proteins, and even post-translation modifications. So we can eventually not look at the bulk contact map of the mixed cell population, but also divide groups of cells into their states and analyze the topology. So at the moment in the lab, we explore mostly two extremes of biology, as I call it, at least of mammalian biology. And one is to look in early development as the cell of the embryo divides. We know from high-C data that the TADs emerge around the blastocyst stage. But for example, at this moment, we don't know if in the cells of the epiblast, which undergo or have many different levels of pluripotency transcription factors, whether the topology is the same or different. And this is an area that we are actively working with the GAM technology to explore in terms also of cell states. However, for today's talk, I'm going to focus on the other extreme of biology, and this is where we look at terminally differentiated cells, in particular cells in the brain. And here the challenge is that we have a very complex tissue. We don't particularly want to extract these cells from their environment and alter their physiological state. But we also want to have maps that are highly cell type specific. And so here the GAM technology is ideal because we can essentially, we have our slice, we get the piece of tissue without disturbing it. We then label at this time in a biased manner the cell of interest using antibodies or morphology or a combination of both. And then once we know within the cell slice which cells we're interested, we can laser microdissect them. And then the pipeline is exactly the same, even though we're now working with brain tissues. And for the purpose of an exploration to ask how different are the topologies of the genome mapped by GAM in specialized cell types, we took 3 brain cell types, oligodendroglia, which we did in collaboration with Gonçalo, and in particular Mandy Major. We visit our lab to learn and collect the oligodendroglia data. We also collected pyramidal glutamatergic neurons. This was from the CA1, and this was work mostly by Isabella Arabula in the lab. And we worked also on dopaminergic neurons. This was mostly Warren Winnicknig from the lab. And we integrated the data with embryonic stem cells, which we had collected previously. And this was work by Robert Bigri. And so we collected this data. So the first thing is really how different are these maps. And I just show you one example where we look at the protocadherin locus on chromosome 18. We're looking at the 3 mega region, and in ES cells you see perhaps the typical TAD organization that we are so used to seeing in many beautiful studies so far. As we look at the brain cells, we understood, and here I should say that only the protocadherin gamma genes are active. As we look at the same locus in the brain cells, we now start to see that we have genes within the locus being active in all the brain cells to different properties. But nevertheless, we see that now with this activity of alpha and beta, that we find contacts between the alpha and the beta, the alpha and the gamma, and the beta and the gamma. And this is true in all the brain cell types, coinciding with the activation of these different genes. But interestingly, this is really just a nuance. We could see also, for example, here in the oligodendroglia, where we have FGF1 becoming active, but not really so much in the other brain cell types, that we now have a specific contact established between the alpha, beta, and gamma loci with the FGF, actually jumping between other genes that are active and, for example, not contacting active genes that are even closer in the linear genome, but are not, for some reason that we don't know so far, chosen for this specific interaction. And so this was just a first example to show you the type of differences that we see. Now, importantly, because we can use so few cells and we need so little material, we can produce these maps from single animals, or even potentially we could use different hemispheres of the brain from the same animal. And we could validate these results and most of the other results that or all of the other results that I'm going to show you. And essentially to see that this is also true, for example, here in littermates of the CA1, but in particular here also in the VTA, where we're using different mice, even with different genotypes. Again, the structure is recapitulated. Now we moved on to analyzing the data genome-wide. And so one of the first things that we all do is to look at the position of tag borders. And so this is what we did. And we essentially use the insulation square method to calculate insulation scores at using different insulation square sizes. And these are the maps that you see under the matrices that I will show in other parts of the talk. Here we're looking at an important region of the genome containing several genes that are affected in neurodevelopment disorders or neurodegenerative disorders. And this is SCN2 alpha, for example, which is most highly expressed in the PGNs here. And it's, for example, associated with autism, epilepsy, and seizures, and different mutations in this locus actually give rise to different diseases with different onset. Extremely complicated to understand how its regulation occurs. Another example is SCN3A alpha here, highly expressed now in DNs and the Parkinson risk gene, and etc. We have a number of other genes also with this type of phenotype in this region. Now, you've been probably looking at the matrices, so you can, for example, look here in the ES cells where these genes are mostly not expressed, that we have a domain of close contacts. As we look in the oligodendroglia, for example, we have the appearance of a very clear loss of insulation, and the appearance of a very clear tag border. So we computed this genome-wide. We chose to use 500 kb insulation square. And initially we did a pairwise comparison, and we found that if we compare every pair of these data sets against each other, that between 25 to 90% of the TADs are changing between cell type, and this is according to the literature. However, we then decided to take all the borders that were found in any of these data sets and do a multi-way comparison, which is shown here as an upset plot. And interestingly, and perhaps to our surprise, we found that actually only 14% of the borders are actually common to all these 4 cell types. That the most abundant borders are actually specific of ES cells, of the 2 oligodendroglia, PGNs or DNs, or common to all brain cell types, or only common to the 2 neurons, PGNs and DNs. And this really suggests that there's much more extensive reorganization of the TADs than perhaps has been appreciated before, partially, we think, because of the pairwise comparisons. And we so decided actually to focus on these most cell type specific borders and ask, as we see here, are there any interesting genes, and in particular genes important for the respective cell types that appear in the borders? And indeed we find so. And I show here only 2 examples just for the sake of time and simplicity. And for example, in oligodendroglia, we find genes important for synapse organization, receptor clustering, fatty acid biosynthesis, whereas in the PGNs, we found genes important, for example, for cognition or membrane depolarization, which are really terms that are really very highly specific of the biology of the cells that we are studying. And this really suggests that as the cells establish their gene expression programs, that in the TAD borders, not only we found what, if we then perhaps a different comparison, what could be more housekeeping genes, but actually genes that are highly cell type specific and highly expressed in any one of these cell types, they actually, their activity coincides with the appearance of these borders, indicating that many of these genes are actually in between TADs, or their activation leads to the loss of contacts and the opening up of chromatin, which perhaps we are more in line with other literature that we are aware. Now, we kept looking at these issues of loss of contacts, and in particular, we got very excited with what we could find at very long genes. And so neurons, many long genes are have this very complex expression in specific cell types, in particular in neurons. And here I have two other examples. One is the GRIC2 gene, a long gene, glutamate receptor, again involved now in epilepsy and cognitive disability. And here we're looking at TS cells and PGNs. And as you can see that this region is mostly devoid of accessible chromatin or activity, very compact in ES cells. And as we look in the PGNs, where the gene is most highly expressed, we can see an extensive loss of contact across the whole region, but in particular, overall in the region surrounding and on the gene itself. A different example now in DNs. Again, you can see that not a lot is going on in terms of expression or open chromatin accessibility in the gene, in the ES cells. As the gene becomes active, extensive gain of open chromatin accessibility, not in particular throughout the coding region, expression and massive loss of these domains. Now we could see this decondensation in our insulation maps that are taken using different insulation boxes. And we were interested to know whether there was a preference for the loss of insulation more at the promoter or throughout the coding region or at the end. And we also wanted to know in a more unbiased way whether this was happening equally to the same extent in the different brain cell types and also in ES cells where long genes or some long genes are also expressed. And so as a first exploration, we took the most expressed genes in each cell type or the least expressed, and we essentially mapped the insulation across the coding region, starting from the TSS all the way from the TS. And we could indeed see that the most expressed genes here in the darker line, that they typically, the all genes, the all coding region tends to lose insulation. And this is occurring at the promoter, but in particular, it's most obvious at the end of the gene where the loss of the insulation is most striking. And this is happening clearly at the PGNs and the DNs, which are the terminally differentiated neurons. In contrast, and interestingly, it's not so obvious at the oligodendroglia, where we also find genes that are decondensing, but there's not this correlation between being highly expressed or least expressed and the events, and also not true or not clear at the ES cells, perhaps only slightly at the promoter region. Now, we wanted to look at these events genome-wide and biasly, and so Dominic Szabo in the lab developed in collaboration with other colleagues in the team a pipeline to essentially quantify this difference in insulation between the maps, and with a statistical test we essentially have a p-value for the difference between the insulation in ES and the brain cell type of interest. We then get a p-value, and we use this p-value to calculate what we call a domain melting score. And with this approach, we could then identify all the genes in each cell type that are undergoing these events. And we can see that we find different genes with the highest melting score in different cell types, although some genes can melt in more than one cell type. You can see RBFOX here, and as well in the oligodendroglia. We, in particular, we then wanted to know, does this somehow relate with expression? And so we compared the melting score with whether the gene expression of genes that melt here in the darker, in the color, and genes that don't melt in gray for in the same cell type. And we could see indeed that in the two brain cell types, the melting coincides with the gene more likely being highly expressed, in particular if the melting is very extensive, as you can see in this likelihood of being more melted to have a higher expression. In contrast, in the oligodendroglia, the melting is also very clearly occurring. We have high melting scores. And there's only then there a slight correlation with the more highly expression. And at this moment, we don't know if this is because of these two being neurons, and here we have oligodendroglia, something to do with their cell type, or actually something to do with the time from which the cells last divided. And so the neurons have been, the last division would have been around birth, so several weeks, a couple of months, which is the age of these animals, whereas the oligodendroglia would have been still dividing a few weeks earlier. And Gonçalo will correct me and explain exactly when they were divided with more precision. So just to give you, importantly, because we have the data from two different animals, and in the case of the DNs, actually from very different animals, even with different genotypes, we compared the melting score of genes from different cell types, and we could see, as you can observe here, that there's really a nice correlation with the melting score itself being highly correlated or extensively correlated to cell types. Nevertheless, there are genes that we find melting only in one of the animals, and this could also be interesting in the context of understanding individual mechanisms of gene regulation and how many of these diseases actually have different penetrance in different individuals that carry the disease. So here we can then conclude that in particular long neuronal genes, when they melt, they actually often highly expressed or with the high chromatin accessibility, which I didn't show you here, but essentially we have very similar maps. We can have genes that are melting with very high expression and not perhaps so much high chromatin accessibility. We can also have genes that melt with high chromatin accessibility without us seeing the mRNA expression that is used in this data. And so there's other details that we are currently exploring in this analysis. And so just to show you just a couple more examples, so you can see that the melting is also not always exactly the same thing. Here is another gene now important in multiple sclerosis, and we can see again highly condensed in ES cells and partially decondensing in oligodendroglia. Sometimes you have almost all gene decondensing. Here in the case of RBFOX, we have generally, there's less contacts, but it's in particular actually in the promoter of the gene that we see this loss of contacts. Now, this was a genome-wide analysis. It's all driven from these matrices and in particular from quantitative analysis of the insulation maps. And so we wanted to go back to single cell biology and actually look at these events with an imaging method. And so we took the RBFOX locus that I was just showing you in the previous slide. Here we have the expression. You can see in ES cells not expressed, highly compact and condensed. As the gene becomes active, we can see that this extensive decondensation, and we wanted in particular to see how does this look if we do imaging. And so this imaging was the work of Isabella Arabula with help from Silvia Carvalho in the lab, and they did FISH, and so taking a number of back probes that cover the all 1.1 megabases of the RBFOX gene. Isa could see that actually in ES cells we have a very clear focus, as it's typical when most of us do FISH. I should say that we're using in this analysis the same tissues that we used for GAM, because we take just so little of the material. You can go back and use the same actually sample, the same biological sample, to do other experiments, and FISH is one of them. When we looked at the locus now in PGNs on the same slices, we could indeed, or ISA could indeed immediately see these very clear events of decondensation. And we had a lot of fun with her sharing these pictures in WhatsApp when she was first collecting them on the microscope. And so this is just one example. You can see the scale is 1 micrometer, and we see this event where the locus is decondensing. Now, Isa went on to quantify this, and you can see that the area micrometer square that occupied by in the mouse ES cells, and clearly in the PGNs, a vast majority of loci is increasing their area, even though we're cutting slices, so we're probably not seeing always the whole locus. And we also notice, and maybe you also noticed looking at these pictures, that in ES cells, the locus is typically at the periphery of nucleolus or at the lamina. And whereas in the PGNs, this was not often the case. And so Isa went on to quantify this in all the loci that she analyzed, and she could see that indeed in ES cells, the locus is most often at the nucleolar periphery or at the lamina, whereas in PGNs, the vast majority of loci are actually in the interior, away from nucleolus and away from the nuclear periphery. Now we took another approach now to have more an understanding of how this melting looks in 3D. And this, for this, we collaborated with the lab of Mario Nicodemi. And here we took another example. Now the neuronexin 3 gene, which is again a very long gene, encodes neuronexin 3, a cell adhesion molecule, which is dysregulated in autism spectrum disorder and schizophrenia. And it's also, I don't have it in this slide, but it's also one of the third most downregulated gene when neurons are treated in vitro with topoisomerase inhibitor. And Mario's lab then produced from the matrices ensembles of polymer models where we can see in the different colors, different parts of the genome. And again, perhaps you were looking already, but if we look, for example, in the green domain here, which you can see is extensively quite compact in the ES cells, but extensively decondensed in the dopaminergic neurons, at least looking at the matrices. And so when we look at these regions in the models, we can again see extensive decondensation, and we can also look at the gyration radius of the green domain in the DNs and see that is extensively decondensed. And I just checked my clock, I'm running out of time. So now I just wanted to use these models to ask us to think about really how we move on from here. So when we think about these structures, we could go into a lot of detail trying to describe every little aspect of every little part of the domain. However, I would argue for discussion that really as a priority, what perhaps we could focus on first is in asking of all aspects, all details of this folding, which are the most important parts that actually drive the folding? What are the bits that are actively contacting or actively avoiding contact? And can we then find what these regions are, zoom in and see what kind of molecular processes are going on in those regions, and then have candidate drivers of the folding and focus first on getting to this. So the questions then are, what are the factors that establish these structures? Which, let's not try to explain everything, which genomic regions do these factors act on? And if we can get to this point, the most crucial points of the topology, can we find out which genes are exactly living there, which ones are directly affected by these mechanisms, and also start studying the bystander effects of the nearby genes. So maybe I skip this for time, or maybe I just say it very briefly. And so we know from thermodynamic principles that you can control the folding of a polymer, and this I should say is work with and from Mario Nicodemi's lab. We can control folding of polymers, in particular chromatin, for example, by increasing the concentration of factors, which even though they're coming in and out, they're highly dynamic because there's so many of them. If one binds, when that one leaves, another one has bound. And the structure is not the same, but it's kept by this process of continuous binding and preservation of the structure. This could also be done by altering the number of binding sites that you have within the region of contact, or by changing the affinity, for example, the histone marks. And so we wanted to start probing these concepts with our data. And to do this, we took now the very closely related, or not closely related, the two neurons, or more closely than, for example, the neurons with the ES. And we wanted to go in and really ask, if we look at these matrices, you can immediately see regions that are different. And if we can focus on the most different contacts, to do this, we essentially subtracted the matrices and focused our attention on only the top 5% most differential contacts. We then combined these most differential contacts with the tag-sec peaks that we obtained. And this was work of Luna Zé Redondo, not only collecting data, but also analyzing extensively the single cell data. And we then asked in particular whether for these tag-sec peaks, do we see putative binding sites for the transcription factors that are differentially expressed between the two neurons. And I need to run now just to say that we did find contacts that contain very differentially expressed TFs that are found within the tag-sec peaks in the cell type. But perhaps even more interestingly, within those contacts that are enriched for those specific TFs that are differentially expressed, we also find a number of upregulated genes. Essentially, half of all upregulated genes in the PGNs are actually found in these differential contacts. And when we look at the gene ontology, again, we find very important terms such as synaptic plasticity, glutamate receptor, which are core to the function of these cells. And I should not finish without saying that these contacts extend very long distances. These are across, in this particular example, 3 megabase regions involving 2 upregulated genes in the PGNs, which establish a hub together with other neighboring genes that are not differentially expressed. Now, we did this in the PGNs, also in the DNs, and so briefly, in PGNs, we found hubs of synaptic plasticity genes. In the DNs, we found, interestingly, genes that are involved in addiction pathways, which are core to the function of dopaminergic neurons. Now, I'll skip through the analysis of compartments, just to say that we're very excited by finding hubs of very long range hubs. This is now 60 megabase regions of sensory receptor genes that form these hubs in the brain cells, but not in the ES, although they're repressed and silent in all of these cell types. And just finally, to summarize, we show extensive specialization of genome architecture. We discover the melting of long neuronal genes, hubs of neuronal specific genes, and also stricter B-compartmentalization of sensory receptor genes in brain cells. Just to finish, I mentioned the names of the major contributors, Warren, Isa, and I should say Mandy also for collecting the data, and Sasha, I didn't mention his name. He did a lot of the genomic analysis as well as the computational biology. Luna, Dominic, and then many other colleagues in the lab have contributed in different ways. And I thank all the collaborators for sharing samples, for doing analysis with us, and all our funders, and you for your attention. And if there is any time for any questions, I'm happy to answer them. Thank you.
Okay, thank you very much, Anna. So please put the questions in the Q&A box. Actually, I'm going to start myself with one question, and it's regarding, so I guess if you would like analyze now in disease, would you, I mean, you had like an example, for instance, of a gene that was decondensed in oligonucleotides and was condensed in ES cells. But I guess this was during normal development. But I guess in disease, perhaps you would expect the opposite. And I was wondering if, for instance, SNPs that might be located might actually have a role on this process.
Yeah, no, absolutely. So this is really, actually, it was at the root of this project. We were initially comparing each of these cell types with actually disease aspects of or environmental treatments. Indeed, we also find interesting differences. So, for example, in the PGNs, if we look in a Down syndrome model or in other mutants such as ATRX of SATB2, we can see changes in the topology. And we think that with this type of analysis, we can essentially understand changes in topology that may not immediately be understood from the more local analysis of gene expression or regulatory regions, which may nevertheless explain why a gene is too much activated, or not activated enough in the mechanism of disease. Also, of course, in differentiation and development, it would be, yeah, it's something that we're very keen to analyze.
Okay, great. And I think because of interest of time, I think we need